Hello everyone! Welcome to my presentation, Osteosarcomas. My name is Chase Gormley and I will be presenting this PowerPoint today. My interest in osteosarcomas actually first started at work. I'm a radiologic technologist at Harborview Medical Center, and I had a patient come in with this pathology, and she was able to tell me a little bit about it and what her doctors had told her. And from then I was really interested, and this project kind of came on good timing with everything, so I got to learn a lot more about this disease. So let's go ahead and begin. To start, osteosarcomas are actually relatively rare. They affect on average about 3 million, or 3 per million per year. So average of 3.4, but we'll round that down to 3. The most affected by this disease include children and adolescents, specifically 10 to 19 being at the highest risk of metastasis to other areas of the body, such as the lungs and pelvis. Survival rates have improved as technology has advanced, and depending on the treatment option most applicable to the case, chances of 5-year survival can be as high as 70% with surgical excision alone. Radiotherapy also exists as a possible treatment option, but it's optimally paired with surgery to improve long-term chances of survival. Next, I'd like to briefly run through the histology of osteosarcomas. Alrighty, a little bit about the histology of these tumors. These tumors stem from mesenchymal cells, which are the cells responsible for differentiating into bone, cartilage, fat, and muscle cells down the line. Originally, it was thought that the tumor cells originated from the preosteoblast and osteoblast cells, which are kind of like the baby bone cells. But further research has more accurately countered that earlier in the cell's lineage, all the way up the family tree there, mutations can occur at the base mesenchymal cell that can alter the karyotype of bone-forming cells sooner and thus result in more established mutations and irreparable damage to the osteoid-producing cells. The event in the mesenchymal cell was introduced in class as chromothripsis, an event that creates a cascade of events that can result in 10 to hundreds of genetic mutations. Speaking of mutations, several hallmarks of mutation have been observed in patients who presented with osteosarcomas or OSs. These include inactivation of the RB1 gene, TP53 mutations, and P53 pregermal mutations. The RB1 gene provides instructions for making a protein called PRB, and this protein acts as a tumor suppressor, which means that it regulates cell growth and keeps cells from dividing too fast or in an uncontrollable way. TP53 is the gene responsible for creating the protein tumor suppressor P53. This guy has been a huge player in our class this quarter, and it has been observed in patients who had osteosarcomas as being mutated or in an inordinate amount. Here are some of the typical cancer pathways that have been observed in OSs. Firstly, DNA methylation. DNA methylation refers to the addition of a methyl group into cytosine on the cytosine phosphate guanine rung, so that CG rung on the DNA. Gene promoters contain dense clusters of these groups, and it has been observed that the levels of methylation are inversely correlated to gene expression. So to be frank, the tumor suppressors are silenced in areas that are hypermethylated. Histone modification acts as another pathway due to its fundamental role in gene expression. It's safe to say that an alteration in one of the two essential building blocks of DNA can't be good. Changes in the histone can lead to inexpression of the RB1 gene that we talked about earlier as the tumor suppressor, but seems to act as the driver for tumor suppression in most cases. Since RB1 can also regulate chromatin structure, further mistakes can be made and mutations can worsen. Nucleosome remodeling is referring to the other essential building block of DNA. The nucleosome is what wraps around the 8 histones and forms DNA in a spool, so like kind of chains around beads, if you're looking at it that way. Changes to the nucleosome directly impact gene expression because they have yet to be translated by RNA and thus have a greater chance of being replicated and mutated due to errors early on. Finally, RNA-mediated events refer to certain copy number alterations in what's called long non-coding RNA, or LNC RNA, which are segments of 200 or greater, that have been observed in over 80% of osteosarcomas. This is a really wordy way of saying that the more common these long non-coding RNAs are mutated in certain patients, the higher chances they'll have these osteosarcomas. Changes to these chains directly impact gene expression and can mistakenly turn off tumor suppressors. So this is a lot going on, but this is a really great example of one of the metastatic pathways that the osteosarcoma may use to migrate and invade other parts of the body. In this case, we see in the right-hand corner there is metastasizing to the lung. Here we see in the left-hand corner here, we're going to go left to right, the spooled chromosome with the labeled structural rearrangement, that little red blast up in the corner. The chromosome is unwound to show the chromatin and histones as separate entities, and we see now the histone is modified and translated by that LNC RNA that we talked about earlier. Studies have also shown that earlier the DNA hypermethylation can lead to gene silencing, in this case silence of the gene P53, or protein P53, which has been observed in over 70% of osteosarcoma cells and 47% of tumor specimens. This is really interesting because the previously stated gene RB1 is also shown to be silenced by hypermethylation in DNA. So the fact that both of these traits are silenced at the same time in many cases of osteosarcomas leads us to believe that hypermethylation directly impacts the dysregulation of both genes and thus leads to a cancer-promoting environment. And so now we're in the center. After the LNC RNA codes that mutation and sends it out of the cell, it now has the blueprints it needs to survive unregulated by the suppressors. It leaves other cells open to invasion and overcrowding, opens up the gates to metastasis to other parts of the body where they can be more harmful, in this case most commonly the lungs and in some cases the pelvis. Two options of treatment exist and are usually used in tandem. Firstly, we'll discuss radiotherapy. Chemotherapy is usually avoided if at all possible after surgery if the tumor remains low grade. However, chemotherapy may be used to halt the progression of the tumor or attempt to shrink it to prepare for surgery. Typically, tumor response to the treatment dictates the survival rate post-operation. Merrill et al. Reviewed that those who had greater than 90% tumor necrosis had an 82% 5-year survival rate, while those that had less than 90% tumor necrosis had a 68% 5-year survival rate. With any treatment option comes risks. Second down there, the risk of infection with any invasive treatment is always something to consider. And due to the already compromised immunity of these patients, this option should be heavily weighed. I also found it interesting that keeping up the dose intensity of treatments was especially important and directly impacted the chances of long-term survival. Dose intensity is basically meaning that you don't want to stop between treatments. So if it's already, you're keeping up with your treatments and you go in for an operation, you don't want to wait too long. So a study conducted by Imran et al. In 2017 concluded that patients who waited over 21 days after surgery to resume chemotherapy had a significantly higher mortality rate. And they suggest restarting chemotherapy within the first 21 days post-op in order to maintain that dose intensity and increase your chances of killing that tumor. So we'll move over to surgery. The goal with surgery is to completely remove the tumor and a generous portion of its border, which is what we're talking about, excision. The tricky part is that it could be highly involved in the joint space, since a large amount of these tumors stem from growth plates on the long bones. When you're thinking about like these patients' populations 10 to 19, all of them have these growth plates on their diaphysis. They're just going right into the joint. They're very close. So the more involved the tumor is in the joint, the less likely that complete excision is possible without amputation. Reconstruction techniques such as endoprosthesis replacement and biological replacement are both viable options and can be assessed as promising course of action. Both are usually more cosmetically pleasing, obviously, to patients than compared to a rotationplasty or an amputation. Right now, titanium prosthetics are the highest quality and are less likely to be infected. Biological reconstructions can be tailored to each patient and can be in the form of allografts or tissue graft. And a combination of both allografts and prosthesis is very common for weight-bearing bones such as the hip or the knee. This is just a really cool picture of different patients who have osteochondromas here on the left, or excuse me, osteosarcoma here on the left. And then in the middle, you see that knee replacement that they've put in post-op. And then all the way on the right, they have an actual hip replacement, a little bit of pelvis hardware as well for 6 years after. So now we come to my favorite part of the presentation, the future and clinical trials. Many options targeting the metastasis of osteosarcomas are being investigated, including my personal favorite, the effect of liquid nitrogen against tumor cells. Proposed by Tushiya et al. At Department of Orthopedic Surgery, Graduate School of Medical Science in Kanazawa University, Japan, The team has been studying the effects of killing tumor cells with liquid nitrogen and replacing the patient tissue after treatment, or reimplanting, I should say, after treatment. Of 17 treated with the frozen pedicle autograft, 14 were completely cured, 2 locally reoccurred, and 1 led to metastasis, unfortunately. Just as well, studies at the Department of Pathobiology at the University of Pennsylvania School of Veterinary Medicine have been using dogs inoculated with an attenuated Listeria vaccine and have been turning up positive results regarding the metastasis and survival of test subjects with OSs. Thirdly, tyrosine kinase inhibitors are currently being explored and are proving to reduce motility, colony formation, invasiveness of disease, and may be beneficial in managing cases of OS metastasis. Another interesting thing that seems to keep returning is good old p53 gene. Researchers seem to agree that a larger test population is needed to further understand and learn about p53's seemingly large role in the formation of osteosarcomas. It has been named an effective biomarker for osteosarcoma prevalence and lung metastasis survival when it's present in its unmutated form. Some clinical trials currently being conducted and further researched include the effect of anti-angiogenic therapy using endostatin. The Department of Orthopedic Surgery in Sapporo Medical University in Hokkaido, Japan, has conducted trials with groups treated with the endostatin therapy and reported a decreased incidence of metastasis and an increase in epithelial growth factor, which is an essential regulator of angiogenesis. However, overall survival was not affected and further research is needed. Another trial, more relevant to our course material, is an exploration of chemokines and chemokine receptors. Specifically, certain chemokine receptors can determine the metastatic destination of tumor cells, which I think is just wild. By tracking the estimated RNA expression levels of these specific receptors, researchers were able to conclude that in a test group of 48 patients, the 2 markers tested showed up 63% and 37% respectively. The levels were inversely related to overall survival and metastasis-free survival. Another group was given an antagonistic agent to these receptors, and 50% of these test subjects showed a marked reduction in microstatic disease. To sum things up, antagonistic agents were added to reduce the incidence of the marked chemokine receptors and reduced metastasis on a microscopic scale. In conclusion, survival rates are dictated by response to therapy, and rates have been improving steadily with advancements in technology and newer resources available today. Clinical trials and new treatments are emerging as more and more progressive discoveries are made, and many reconstructive routes for surgery exist today. Not only the prosthesis and the biological reconstruction, but as we get further and further into, I think, stem cell research, when they can actually start like growing larger samples of people's tissue, I think we may be able to grow sections of bone instead of having to worry about replanting already, donor bone that might, it's called a non-union, but it might not form to the bone correctly, and then they have to go back for surgery. We see that a lot at work. P53 and RB1 gene expression could also be the key to future research of metastasis. Of all the research I did for this project, which was actually, there was a lot of P53 and RB1 gene expression all over the place. It was mentioned multiple times in multiple summaries and conclusions, and I really think it's going to be the key to figuring out the metastasis of this disease and improving the survival rate of patients in the future. Thank you.